Recombinant DNA technology and biotechnology. So let's have an introduction about this. Recombinant DNA technology, also known as genetic engineering, molecular biology technology, or cloning, is a set of techniques that enable DNA from different sources to be identified, isolated, and recombined so that new characteristics can be introduced into an organism. One of the main technique in recombinant DNA technology is DNA cloning because it produces an unlimited number of copies of a particular DNA segment. The recombinant DNA technique was engineered by Stanley Norman Cohen and Herbert Bayer in 1973. What is DNA cloning? DNA cloning is the production of large number of identical DNA molecules from a single ancestral DNA molecule. The essential characteristics of DNA cloning is that the desired DNA fragments must be selectively amplified, resulting in a large increase in copy number of selected DNA sequences. In practice, this involves multiple rounds of DNA replication catalyzed by a DNA polymerase acting on one or more types of template DNA molecules. Essentially, two different DNA cloning approaches are used, like cell-based DNA cloning. This was the first form of DNA cloning to be developed and is an in vivo cloning method. The first step in this approach involves attaching foreign DNA fragments in vitro into DNA sequences which are capable of independent replication. The recombinant DNA fragments are then transferred into suitable host cells where they can be propagated selectively. Then cell-free DNA cloning. The polymerase chain reaction (PCR) is a newer form of DNA cloning which is enzyme mediated and is conducted entirely in vitro. The essence of cell-based DNA cloning involves following steps. Cell-based DNA cloning requires attachment of DNA fragments to purified replicons in vitro condition and propagation in suitable host cells. Construction of recombinant DNA molecules. Recombinants are hybrid DNA molecules consisting of autonomously replicating DNA segment plus inserted elements. Such hybrid molecules are also called chimera. Construction of recombinant DNA molecules by in vitro covalent attachment, ligation of the desired DNA fragments target DNA to a replicon, any sequence capable of independent DNA replication. This step is facilitated by cutting the target DNA and replicon molecule with specific restriction endonucleases before joining the different DNA fragments using the enzyme DNA ligase. The recombinant DNA molecules are transferred to host cells, often bacterial or yeast cells, in which the chosen replicon can undergo DNA replication independently of the host cell chromosomes. Then selective propagation of cell clones. Selective propagation of cell clones involve two stages. Initially, the transformed cells are plated out by spreading on an agar plate in order to encourage the growth of well-separated cell colonies. These are cell clones, populations of identical cells, all descended from a single cell. Subsequently, individual colonies can be picked from a plate and the cell can be further expanded in liquid culture. Isolation of recombinant DNA clones. Isolation of recombinant DNA clones by harvesting expanded cell cultures and selectively isolating the recombinant DNA. Cloning vector is here, target DNA is there, recombinant and another non-recombinant is also produced. Introduction of cloning vector into host. Bacterial chromosome is there, non-transformed. Selection of transformed cells with selection of recombinant. Then later here selection of recombinant and the selection of transformed cells can be done with the help of antibacterial, antibiotic resistance, that kind of properties which we'll be learning later. So this amplification is the last step. This shows an overview of DNA cloning bacteria using a plasmid vector. So tools of recombinant DNA technology. Enzymes for DNA manipulation. The enzymes used in the molecular biology fall into four broad categories. Template dependent DNA polymerase. DNA polymerase enzymes that synthesize new polynucleotide complementary to an existing DNA or RNA template. Different type of DNA polymerase used in gene manipulation. DNA polymerase 1. Conberg polymerase has both 3' 5' and 5' 3' endonuclease activities and 5' 3' polymerase activity. Reverse transcriptase, also known as RNA-directed DNA polymerase, synthesizes DNA from RNA. Reverse transcriptase was discovered by Howard Temin at the University of Wisconsin and independently by David Baltimore at about the same time. The two shared the 1975 Nobel Prize in Physiology or Medicine. Taq DNA polymerase. DNA polymerase derived from a thermostable bacterium, Thermus aquaticus, operates at 72 degrees Celsius and is reasonably stable above 90 degrees Celsius and used in PCR. Taq DNA polymerase, which is derived from Thermus aquaticus. So, template dependent DNA polymerase, enzymes in DNA manipulation, DNA polymerase 1, reverse transcriptase, Taq DNA polymerase. And nucleases are needed, enzymes which degrade DNA molecules by breaking the phosphodiester bonds that link one nucleotide to the next. There are two different kinds of nuclease. Exonuclease remove nucleotide one at a time from the end of a DNA molecule. Endonucleases are able to break internal phosphodiester bonds within a DNA molecule. Then restriction enzyme or restriction endonuclease, discovered by Werner Arber, Daniel Nathans, and Hamilton Smith, for which they received the 1978 Nobel Prize in Medicine. A bacterial enzyme cuts double-stranded DNA by cleaving the phosphodiester bonds. The term restriction comes from the fact that these enzymes restrict the entry of foreign DNA in the bacteria. Restriction enzymes, therefore, are believed to be evolved by bacteria to resist viral attack. And special characteristics of restriction enzymes have certain unique features that make them indispensable in recombinant DNA technology and biotechnology. These characteristics are listed below. Restriction site sequences recognized by most restriction enzymes are palindromes. The restriction site sequences recognized by most restriction enzymes are palindromes. Most restriction enzymes are highly specialized to such an extent that they recognize only a single restriction site. The restriction site is recognized irrespective of the DNA source. Nomenclature. The name of any restriction endonuclease consists of four parts. The letter R or M to designate restriction endonuclease or methyltransferase respectively. An abbreviation of the genus and species of organism to three letters. A letter of R or M to designate restriction endonuclease or methyltransferase respectively. So the letter R for restriction endonuclease and M for methyltransferase. M is also used. So an abbreviation of the genus and species of the organism to three letters, like ECO for Escherichia coli, identified by the first letter of the genus name and the first two letters of the specific epithet. A letter, number or combination of the two to indicate the strain of the relevant species. Hint, from H. influenzae from strain RD. Roman numeral to indicate the order in which different restriction modification systems were found in the same organism or strain, like DSA 6 was the sixth restriction modification system found in Dactylococcyx salina. And sites of cleavage. Rather than cutting DNA indiscriminately, a restriction enzyme cuts only double helical segments that contain a particular nucleotide sequence known as a recognition sequence. Recognition sequence always in the same way. The recognition sequence for the vast majority of type 2 restriction endonucleases are normally palindromes. That is, the sequence of bases is the same on both strands when read in the 5' to 3' direction as a result of a two-fold axis of symmetry. Restriction enzymes make either blunt or staggered cut. Thus, restriction fragments may have blunt ends. The cleavage points occur exactly on the axis of symmetry. and overhanging ends. The cleavage points do not fall on the symmetry axis so that the resulting restriction fragment process are called 5' overhangs or 3' overhangs. Overhanging ends generated by cleavage with restriction nucleus are often described as sticky ends or cohesive termini because the two overhangs ends of each fragment are complementary in base sequence and will have a tendency to associate with each other or with any other similarly complementary overhang by forming base pairs. Staggered cut can be seen here. 5' staggered ends, 3' staggered ends. Different fragments with the same sequence in their overhanging ends can be generated by cutting with the same restriction nucleus, cutting with different restriction endonucleus that happen to recognize the same target sequence. Cutting with the same restriction nucleus, cutting with a different restriction endonucleases that happen to recognize the same target sequence. Isoschisomers, or by cutting with enzymes that have different recognition sequences but happen to produce compatible sticky ends. Then frequency of recognition sequences. The number of recognition sequences for a particular restriction endonuclease in a DNA molecule is of known length can be calculated mathematically. It is assumed that the nucleotides are ordered in a random fashion and that the 4 different nucleotides are present in equal proportions. Then the restriction site of 6 BP will occur on average every 4 raised to 6 bases. So a array which recognizes the sequence of 6 base pair will produce fragments each of which is an average just over 4 kb. Similarly, enzymes with 8 BP and 4 BP base pair restriction site produce longer 4 raised to 8 BP and smaller 4 raised to 4 BP fragments respectively. And we can predict the sequence shown at one stand given the 5' to 3' direction and the blunt end or sticky end at the end. We can predict that, I guess. Comparative characteristics of 3 types of restriction endonuclease characteristics. Type 1 restriction and modification system, single multifunctional system, protein structure, 3 different. Wait a minute, I'll be reading each point from each section. Characteristics, restriction and modification system. Type 1 single multifunctional system enzyme, then separate endonuclease and methylase, separate enzymes with one common subunit. And protein structure, 3 different subunits, 2 identical subunits, 2 different subunits. A restriction requirement ATP, Mg2 plus and S adenosyl methionine. Then Mg2 plus is the requirement for type 2 ATP and Mg2 plus is the requirement for type 3. ATP is not needed in type 2, that is the point. Then cleavage sites, random, at least 1000 base pair from specific site. In case of type 2, at or near host specific site. Then type 3, 24 to 26 base pair to 3' host specific site. Okay. Then restriction modification systems, bacteriophages that are liberated from bacterium of a particular strain can infect other bacteria of the same strain but not those of a different strain. This is because the phage DNA has the same modification pattern as the DNA of a bacterial strain, so it can infect. The phage is restricted to that strain of bacteria. The restriction is not an absolute one. Some phages can escape restriction and can acquire the modification pattern of the new host. A system of enzymes that methylizes nucleotides at recognition sites and hence protects the recognition sites from restriction enzymes is called a restriction modification system. The restriction modification system involves 2 types of enzyme activity. A sequence-specific DNA methylase activity provides the basis of the modification pattern. Then a sequence-specific restriction endonuclease activity underpins a restriction phenomenon by cleaving phage DNA, whose modification or methylation pattern is different from that of the host cell DNA. And the bacterial strain possesses a DNA methylase activity with the same sequence specificity as the corresponding restriction endonuclease activity. As a result, cellular restriction endonucleases will not cleave the appropriately methylated host cell DNA but may cleave incoming phage DNA if not methylated appropriately. Note, however, that some plasmid and bacteriophages possess genes for modification and restriction systems so that their presence in a bacterium determines its specificity. The mung bean nuclease digests single stranded nucleic acid but will leave intact any region that is which is double helical. The nuclease S1 has mung bean nuclease, however, the enzyme will also cleave a strand opposite a nick on the complementary strand. Then RNA is one RNase A nuclease which digests RNA but not DNA. RNase H nuclease which digests the RNA strand of RNA DNA heteroduplex. And end modification enzymes. End modification enzymes make changes to the ends of DNA molecules. Terminal deoxynucleotidal transferase. So then in the modification enzymes, you are learning end modification enzymes. Terminal deoxynucleotidal transferase. It is a template independent DNA polymerase. It is a template independent DNA polymerase because it is able to synthesize a new DNA polynucleotide without base pairing of the incoming nucleotides to an existing strand of DNA or RNA. This enzyme is used for the formation of cohesive end by homopolymer tailing. A homopolymer is simply a polymer in which all the subunits are the same. Enzyme terminal deoxynucleotidal transferase catalyzes the addition of a series of nucleotides onto the 3' OH termini of a dsDNA molecule. If this reaction is carried out in the presence of just one type of deoxyribonucleotide, a homopolymer tail is produced. So 5' to 3' reduction, terminal deoxynucleotidal transferase DCTP. So here we could see this terminal deoxynucleotidal transferase DCTP is also needed. Then alkaline phosphatase obtained from various sources including E. coli and calf intestinal tissue removes phosphate groups from the 5' end of DNA molecule, which prevents these molecules from being ligated to one another. So alkaline phosphatase. Then T4 polynucleotide kinase. T4 polynucleotide kinase obtained from E. coli cells infected with T4 phage performs a reverse reaction to alkaline phosphatase. T4 polynucleotide kinase obtained from E. coli cells infected with T4 phage performs a reverse reaction to alkaline phosphatase, adding phosphatases to 5' ends. Its main application is in the end labeling of DNA molecules. Okay. Then ligases, DNA ligases join DNA molecules together by synthesizing phosphodiester bonds between DNA ligases join DNA molecules together by synthesizing phosphodiester bonds between nucleotides at the ends of two different molecules or at the two ends of single molecule. DNA ligases commonly used in cloning experiments are those obtained from E. coli or from the bacteriophage T4. Then cloning vectors are needed. An autonomously replicating genetic element used to carry a complementary DNA or fragment of genomic DNA into a host cell for the purpose of DNA cloning. A viral DNA or plasmid can be used as a cloning vector. Cloning vectors for E. coli cloning vectors based on plasmid. Plasmids are naturally occurring circular extrachromosomal autonomously replicating DNA present in many prokaryotic and few eukaryotic organisms. Plasmid vectors provide a simple way of cloning small DNA fragments in bacterial and simple eukaryotic cells. The ideal plasmid cloning vector must have small size, high copy number, own origin of replication, its own. Okay. Then restriction sites for many restriction enzymes and selectable markers. Thus, in order to adapt natural plasmid molecules as cloning vectors, several modifications are normally made. Insertion of a multiple cloning site polylinker. This is a short synthetic sequence which contains unique restriction sites for a variety of common restriction nucleases. Pre-existing restriction sites for these enzymes will be detected from plasmid if necessary to ensure the presence of unique cloning sites. So cloning vectors we are learning. So plasmids we mentioned, insertion of an antibiotic resistance gene, the host cells that are used must naturally be sensitive to the antibiotic in question so that any vector molecule which transforms the host cell can confer antibiotic resistance. By plating transformed cells on a medium containing the antibiotic, only those cells that have been transformed by vector molecules survive. Insertion of a selection system for screening of recombinants. Typically, this involves arranging for the multiple cloning site polylinker to be inserted into an expressible gene or gene fragment within the plasmid. An example of a widely used cloning vector is PBR322, which replicates in E. coli. In PBR322, P stands for plasmid and BR stands for its developer, Bolivar and Rodriguez. Then 322 distinguishes this plasmid from others developed in the same laboratory. It is small, only and maintained in the host and relatively high copy number 20 to 30 copies per cell. It has genes conferring ampicillin resistance and tetracycline resistance on its host and single cleavage sites for PST1, EcoR1, HIND3, BAM1, SAL1, and CLA1. PBR322 is genetically engineered from DNA derived from three different naturally occurring plasmids, R1, R65, and PMB1. Antibiotic resistance genes ampicillin R and tetracycline R are derived from plasmid R1 and R65 respectively. Origin of replication derived from PMB1. The valuable features of PBR322 have been enhanced by the construction of a series of plasmids termed PUC, produced by the University of California. The plasmid vector PUC19 contains a polylinker with unique cloning sites for multiple restriction nuclease and an ampicillin resistance gene to permit identification of transformed cells. In addition, selection for recombinants is achieved by insertional inactivation of a component of a beta galactosidase gene, a complementary portion of this gene being provided by using a specific modified E. coli host cell. Bacterial artificial chromosome or BAC back cloning vectors were developed by Mel Simon and his colleagues. Back vectors are maintained in E. coli as large single copy plasmids and contain insert of 50 to 300 kb. Back vectors contain the F plasmid origin of replication, F plasmid genes that control plasmid replication and plasmid copy number, and the bacterial chloramphenicol acetyl transferase gene for plasmid selection.